right, so thanks so much everyone for coming to my final seminar today, and thanks so much Dimity for the introduction. Today I'm going to be presenting my PhD research, which is titled Weakly Supervised Segmentation of Underwater Imagery. I'd like to start by acknowledging the traditional owners of the land where QUT now stands, the Turrbal and Yagara people, and recognise that these lands have always been places of teaching, research and learning. In this thesis, we also collect a large data set of images in the Moreton Bay region. So I'd also like to recognise the traditional custodians of that region, the Quandamooka people, and recognise them as the custodians of that land and sea country. I'll start my presentation today by describing the motivation and background for the thesis, including the two underwater ecosystems we consider, seagrass meadows and coral reefs. I'll then describe various marine survey methods, and then I'll identify the characteristics of underwater imagery and how these images differ from the typical images that we see in mainstream computer vision tasks. That will lead into a literature review in which I'll cover seagrass and coral datasets and prior approaches, and then I'll identify some weakly supervised segmentation approaches from other domains. I'll then cover the 4 contributions of this thesis. And first, I'll discuss our approach for multi-species seagrass detection and classification from patch labels, our second contribution which uses image level labels, and then contributions 3 and 4 focus on the multi-species coral segmentation task. Finally, I'll identify the impact of this thesis, potential avenues for future work, and then I'll conclude the seminar. So starting now with the motivation and background for this work. So the first coastal ecosystem we consider in this thesis are seagrass meadows, which are comprised of marine flowering plants which grow in the seafloor. Seagrass meadows are important for the range of multifunctional roles which they provide. They're important for supporting fisheries, they act as a nursery for marine species, and they're also an important source of food for dugongs. Seagrass meadows are also critical for their ability to sequester and store large quantities of blue carbon. Marine vegetated habitats, which include seagrass meadows, salt marshes, mangroves and macroalgae, comprise only 0.2% of the entire ocean, but they really punch above their weight in the amount of carbon that they sequester, and they comprise 50% of the total carbon stocks of the ocean. The second underwater ecosystem we consider are coral reefs. Coral reefs support a really rich abundance of marine life and are some of our most complex and biodiverse ecosystems. Coral reefs are also important for their ability to protect coastlines, and they support economies worldwide through tourism and fishing. However, coral reefs are under significant threat due to both natural and anthropogenic impacts, such as increased ocean temperatures due to climate change, increased frequency of damaging tropical cyclones, and predation by the crown of thorns starfish. We perform marine surveys to enable us to map and monitor these marine habitats and track their health over time. Having availability of data at varying spatio-temporal scales is needed to inform active management and active interventions. We need to also monitor our ecosystems for impacts of pollution, climate change, and some of the other factors which I've already mentioned. On the image on the slide, we can see an ecologist performing a manta tow survey. So this is quite a manual survey technique where they're towed along through the ocean and they're noting down species that they see. More recently, we've seen advances in the use of other marine survey methods. So we can see here a whole range of survey methods which have different benefits and limitations in terms of the 2 factors that we can see on the slide. So on the y-axis, we can see spatial scale, which can be interpreted as the distance or the area which that method is able to cover. And on the x-axis, we have the taxonomic resolution, which is the level of detail that you can see in the imagery collected by these approaches. In this thesis, we're interested in focusing in on use of autonomous surface vehicles and autonomous underwater vehicles for the benefits that they provide in terms of broad-scale survey coverage, while still offering a high tax. In this thesis, we do still cover imagery collected by snorkelers, divers, and through photo-quadrat surveys, as a lot of the historical data has been collected using these approaches. So now just a few examples of the approaches that we're considering. So on the left, we can see a remotely operated vehicle, and on the right, one of QUT's floaty boat autonomous surface vehicles. And the inset video there is showing example footage collected by this vehicle. So now I'm going to highlight some of the key differences between underwater imagery and the types of typical computer vision images that we see in more mainstream applications of computer vision. So first of all, these typical images have a clear foreground-background separation. So this means that you'll typically have some sort of object that's in the image that has a clearly defined boundary. These images also have high annotator agreement. So if I asked 5 of you to label the image that we can see here on the bottom of the frame, it's most likely you would all identify it's a human on a motorbike, and you would probably draw a similar segmentation ground truth mask. What this means is that we can use crowdsourcing platforms like Amazon's Mechanical Turk to go ahead and label large quantities of data in quite a cost-effective and fast way. This isn't the same for underwater imagery. So in underwater images, we have huge amount of variation in the image characteristics, and this is both within datasets and between datasets due to a range of factors. So firstly, lighting and depth plays a really significant role. Use of artificial lighting can change the image characteristics. And if the sun is shining through the water, we get these shadows or ripples, which can also change how our images appear. Camera angle and distance also has a role to play. So in the right image, we can see a forward-facing image, and the corals here appear as though they have a plate-like structure. If we take an image directly from the top-down viewpoint, that feature is no longer visible and the corals appear differently. Finally, turbidity and blur play a significant role in underwater imagery and definitely is something we have to contend with. Underwater images are also highly complex, so it's common to have really overlapping instances in these images, and the boundaries are often poorly defined as well, so it's hard to tell where one thing ends and the next thing begins. There's also a clear lack of objectness. So as I mentioned earlier, in typical applications, we have clearly defined objects. Here, we're interested in the seagrass, but it's not a single object in this image. Instead, it's growing as a carpet over the seafloor. So for all of the reasons I've already said, it's important that we have our images labeled by domain experts, but there's also species-specific reasons why we need to do that as well. So firstly, we can have the same species of seagrass that has different visual appearances due to effects like seasonality, the location it's growing in, and also the environmental conditions. The opposite can also be true. So you can have different species of seagrass, or here we have entirely different families of corals that for an untrained observer would appear to look like the same species. So it's really critical that we have domain experts that are labeling images for us. So now I'm going to discuss the literature review for this work, starting with seagrass, and then I'll touch on the approaches for coral. So for seagrass, one of the main data sets that we see in the literature is this looking for seagrass data set, which has been collected by an underwater vehicle, but labelled only with polygons for the presence or absence of seagrass. So it's only a binary data set. And the other data sets that we see frequently are this Edith Cowan University Holophila ovalis data sets 1 and 2. And these data sets are labelled with bounding boxes around each leaf of the seagrass, but only contain one type of seagrass. So from our survey, we identified a clear gap in the availability of multi-species seagrass data, and that's something that we want to address in this work. And this leads into our first research question, which is how can we design data collection methodology in such a way that we can reduce the annotation effort for those domain experts? In terms of prior approaches, there are many approaches which use that densely labelled looking for seagrass dataset and train segmentation models in a fully supervised end-to-end way on that data. And there are also approaches which perform only binary presence-absence detection of seagrass or single species detection as well. So from this, we really identified a need to design deep learning algorithms that can be trained from a weaker supervisory signal. And this is so that we can move away from having these densely labelled ground truth masks when training our model. So now for the coral use case, the UCSD Mosaics dataset is the only densely labelled dataset of coral images which we have available. And so for this reason, we do use this dataset extensively in the third and fourth contributions of this thesis. But we do have a large availability of photo quadrat data. So this is data taken from the top-down viewpoint in a very controlled way. And then the images are labelled using the coral point count methodology. So this involves randomly distributing or spacing points as a grid in that image, and an ecologist will label only the points that have been specified by the coral point count method. And typically there would be between 100 and 300 points per image. So this leads into our next research question, which looks at how we can design deep learning algorithms which are able to leverage these existing quantities of weak expertly labelled data. So even though this data hasn't been collected with deep learning in mind, how can we still design something that can use this as a really important source of that expert knowledge? There are some existing approaches that aim to do this. So there are many approaches that take the randomly placed point labels and will take a crop around each of those points and use those crops for classification. But the problem with this approach is that, as we can see here, oftentimes the point in the middle of the patch is not representative of the dominant species in that patch. So in this work, we're aiming to move away from these types of approaches. What we are interested in here is the task of point label propagation. So in this work, what we have is an input image and some sparse labels. Here they've used grid-spaced point labels. And what we're wanting to do is propagate those points within our image to obtain what's called an augmented ground truth mask. So we can propagate those points, obtain that mask, and then use it to train a model as usual in that end-to-end way. So how this approach achieves this is by generating superpixels. So a superpixel is a group of pixels that have been grouped together into a segment. And the idea here is that the pixels have some similar features. So in this work, they use color features or RGB values to generate those superpixels. And then they combine the superpixel segments with the sparse labels to obtain the augmented ground truth. But they don't use the points in the superpixel generation itself. They only consider them as a post-processing step. Now the authors take this work a step further and contribute the coral seg work, and this still uses the multi-level superpixel approach, but here they compare a bunch of different segmentation architectures and loss functions, and so we compare our methods to these works. This work also gives us inspiration for our fifth research question, which is how can collaboration between human experts and the deep learning algorithm improve label efficiency? So these approaches all assume you have between 100 and 300 point labels. We're interested in whether we can reduce that required quantity of labels by leveraging human expert knowledge. So this now leads into a discussion on weakly supervised segmentation in other domains. So there are point supervised approaches in the literature, but these approaches all assume that the points have been placed centrally on the objects of interest. This isn't the case for us with our underwater imagery, where the points are either placed as a grid or placed randomly in the image. There are also approaches in the literature that leverage class activation maps. So these methods take feature maps from the final layers of a convolutional neural network and use that as a pseudo-label in performing the weakly supervised semantic segmentation task. All of these approaches have a key assumption of objectness. So this means that there is some clearly defined object in the foreground. In our situation, we don't have that assumption of objectness, so the performance of these approaches is degraded. Over the course of my PhD, there's been significant advances in the use of vision transformers and a real shift towards using larger models trained on more data, which aim to produce more generalized representations. So this informed our research question number 3, which is how can deep learning algorithms leverage pre-trained large foundation or vision language models in really domain-specific applications? So do these models still have worth when we're trying to perform a really specific task? So I'll touch on a few of the key advances that I'll use later on in my thesis. So firstly, the large vision language model CLIP. Now this model combines features from natural language prompts and image features in the same embedding space. And here we're interested in whether we can leverage that shared embedding space for a domain-specific task. And I'll touch more on this shortly. Another key advancement in the last few years was the segment anything model, or SAM. Now, this model aims to perform instant segmentation in any image, and the authors argue that their model has a really robust, generalized idea of what an object is. So just to demonstrate that this model doesn't solve the problems in underwater imagery, we tried it on seagrass images from 2 data sets, from Floaty Boat and from the Global Wetlands data set, and we've also tried it on coral images as well, and show that this approach doesn't produce useful segments in these style of imagery. Another noteworthy model is the Dino V2 model. Now, this aims to produce really robust task-agnostic features. So with this model, we aim to use it in our 4th contribution and see whether these features are useful in domain-specific tasks. So now I'm going to discuss the 4 main contributions of this thesis, starting with our first work, multi-species seagrass detection and classification from underwater images. And this work looks at how we can design the data collection methodology in such a way to reduce annotation effort, and we also want to design our deep learning algorithm so that it can be trained from a weaker supervisory signal. So when we have seagrass images, it's very common to have a very cluttered, complex image with many overlapping instances. It's simply too costly and time-consuming for an ecologist to go ahead and provide polygon masks around each of those leaves, or to provide pixel-level annotation. So instead, we propose this new task, which we call coarse seagrass segmentation. And here we're aiming to perform inference as a coarse grid over our image. So we believe that this offers a really good balance between having sufficient resolution such that we can estimate species composition and extents while still reducing the amount of annotation effort for that domain expert. So to this end, we design a dataset collection methodology which transfers the effort from annotation time instead to data collection time. So what this means is that we encourage the data collector to only collect images which contain just one species of seagrass at a time. And this means that we can take all of the patches in each image and label them as the same class as the image level label. And in this way, we can really quickly collect a large data set of multi-species images. So we contribute the deep seagrass dataset, which is a large dataset across 3 different types of seagrasses and also a background class. We then train a patch classifier on this data. So we use a simple VGG16 classifier, and we train it end-to-end on those patches. And then at inference time, we can apply it as a strided sliding window across that image to perform our coarse segmentation task. And here we tried 2 different classifiers, and we can see that we achieved 87% on our test dataset. Some qualitative results for our work. So we can see the patch level classifications are shown using the colors, whereas we also see the image level labels there as well. In terms of failure cases, what we see most commonly is that the edge patches or the blurry or visually degraded patches are most commonly misclassified with this approach, and this is something that we tackle in the 2nd contribution of this thesis. So now, just to summarize this first contribution, we proposed the first approach for multi-species classification of seagrass with deep learning. We contributed a novel semi-automated dataset collection methodology, and to go with that, a large dataset of multi-species seagrass images. And we believe that the approach presented here could be used as a basis for estimating the percentage cover of seagrass species. So this leads into the 2nd contribution of this thesis. So here we're looking at how we can leverage deep learning algorithms trained from a weaker supervisory signal, but we also bring in our research question 3 as well, which looks at how can approaches leverage pre-trained large foundational vision language models in really domain-specific tasks. And in this work, we're aiming to only leverage the image level labels. So in that prior work, we used patch level labels to achieve the coarse seagrass segmentation inference. Only the image level labels, but we still want to achieve the same level of detail and still perform that coarse seagrass segmentation task at inference time. So in this work, we contribute 2 complementary classifiers. The first we call C-feats, and this is based on contrastive training and feature similarity. So first, what we do is we take a ResNet 18 feature extractor and we perform an unsupervised pre-training task, and we use the SimCLR contrastive framework for this task. Once we have our initialized feature extractor, we can go ahead and calculate a set of template feature vectors for each of the classes that we're wanting to classify. So how we do this is by taking our feature vectors from our ResNet 18 feature extractor, and we do this for of n patches, and we go ahead and average those feature vectors Vx, where each of those patches has been labeled at the image level as the class. So for each class, we're averaging a whole bunch of patches that have been labeled at the image level as that class to obtain what we call this template feature vector. Once we have our templates, one for each class, we can go ahead and calculate the cosine similarity between each query patch, Vx, and our template feature vectors for each class. And we can go ahead and use this to create a pseudo label to tell whether that patch is more likely to contain background or seagrass. So this enables us to have pseudo labels at the patches, which we can then use to train our entire architecture end to end. And this means that we can also update those template feature vectors every epoch while we're training. Our second approach is called CCLIP, and this approach uses the CLIP large vision language model as a pseudo label generator. So we take the CLIP model and we query it with a patch and also a whole bunch of natural language prompts. We can then use CLIP to tell us which of those prompts is most likely to correspond with the patch, and this tells us whether the patch contains seagrass or background. So here we're using just the general knowledge of CLIP. If it does contain seagrass, we can propagate the domain expert image level label to that patch and use it as a pseudo label for training the architecture end to end. And we combine these 2 classifiers in an ensemble to get our overall approach. So we can see here that best out of the 2 approaches, but when we combine it with CCLIP, we do see improved performance across the board, and we can inspect qualitative outputs to understand why. So in the bottom left corner of the C-feats approach, we can see those slightly blurry patches, it's actually misclassified the type of seagrass there. Whereas the CCLIP model behaves far more conservatively, and in any of the blurry patches, it's far more likely to classify those patches as background rather than overconfidently classifying it as seagrass. So if different behaviors of these 2 classifiers together, we can get a far more robust performance from our ensemble. So what we can see here in the results is that we've improved on the state of the art from 88.5% to 95.3% with our approach. In this work, we also perform a real-world deployment case study. So here we've taken footage collected by the Floaty Boat autonomous surface vehicle, and we're using our approach as seen here on the slide. So 3, and the yellow color shows classifications of seagrass, and the pink color shows where the model has classified the substrate as background. So in this work, we've contributed a coarse seagrass segmentation approach trained only from image level labels. We contributed 2 complementary classifiers, the first based on contrastive training and feature similarity, and the second which leverages the CLIP large vision language model, and showed that the ensemble improves the performance. And then we performed that real-world case study on imagery from an autonomous surface vehicle. So this leads now into our third contribution, label-aware superpixels for multi-species segmentation of underwater imagery. And in contributions 3 and 4, we're now considering the coral multi-species segmentation task. So here we have images taken using the photo quadrat survey method. They're top-down images, which are then labeled using the coral point count methodology, and in this case, randomly distributed sparse point labels. In traditional semantic segmentation, we have pairs of training images and dense ground truth masks, which are used to train our architecture, whereas in this work, we're taking training images with sparse point labels, and we use these in a point label propagation approach to obtain our augmented ground truth masks, which we then use to train the semantic segmentation architecture, and that model can then be used for inference on unseen images. So prior approaches have taken these photoquadrant images and sparse point labels, but they haven't leveraged the points themselves in the generation of the superpixels. And these approaches also don't use deep features, they're based only from color features. In this work, we use superpixels informed by the points themselves, and this encourages those superpixels to encompass single species regions. Our point label aware superpixel algorithm is comprised of two loss terms. The first is a weighted distortion loss, and this minimizes distances between deep pixel features. And then our second loss term is the conflict loss, and this loss term considers those conflicting point labels and says that where the points have different class labels, they should be placed in different superpixels. And so the combination of these two loss terms produces superpixels that really closely conform to our species boundaries. So here we can see the superpixels at work. So initially, the superpixels are more evenly spaced out in the frame, and then as those superpixel centers are optimized, the superpixels can cluster where the image is more complex or spread out if the image is more simple. So we can see that here we have a really accurate augmented ground truth. So with this approach, we improve on the prior state of the art from 10% to 85%, and we also improve on the computation time per image as well. So in this work, we've contributed a novel approach for generating superpixels based on deep CNN features. We've proposed a novel superpixel algorithm which incorporates the point labels and ensures that the segments encompass only single species regions at a time. And we also propose using deep CNN features as opposed to just the RGB colors. And this enables us to improve the point label propagation accuracy and mean intersection over union while also reducing the computation time as well. So now this leads into our contribution for human-in-the-loop segmentation species coral imagery. Now this work considers 3 of our research questions. In this work, we're still aiming to leverage those existing quantities of point label data. We're also wanting to leverage large pre-trained foundation or vision language models in this work. And then we also introduce our research question 5, which looks at whether collaboration between human experts and the deep learning model together can actually improve our point label propagation, our point label efficiency. So in the previous contribution, we were considering the use case where we had typically between 100 and 300 point labels per image. In this work, we're really interested to see whether we can still have accurate point label propagation, but with far fewer point labels. So here we're considering the extremely sparse label setting, where we have only 5 to 25 point labels per image. So our approach uses the denoise Dino V2 model as a general feature extractor, and we don't need to do any fine-tuning or training of this model. Image with sparse point labels, we use that model to obtain features, and we obtain features for the labeled points and also every pixel in the image. And from this, we can use a k-nearest neighbor algorithm to do clustering in the deep embedding space. And with this clustering, we then obtain our augmented ground truth mask. Since we're considering that extremely sparse point label setting, we also propose a human-in-the-loop labeling regime. So with this regime, we take our input image and we give it to our domain expert and ask them to label up to 10 points centrally in the largest coral instances in the image. Then we take that denoised Dino V2 model to obtain our features, and we do that clustering with the KNN algorithm. And from this, we can obtain this inverted cosine similarity map. So this map shows us the cosine similarity for each pixel to its nearest known labeled pixel. And what this does is it shows us where in the image, potentially the points haven't captured a type of coral, or maybe part of the image has different visual characteristics to the rest of the image. So in those areas, we should have another point label so that we can account for that region. We also create this distance map, and this encourages exploration within the image. And we combine the two maps together and take the maximum as the next pixel for labeling. So we feed this point back to our domain expert, and we can continue this process iteratively until the maximum desired number of point labels is reached. From this, we get our augmented ground truth mask, and we can then use that to train a model to perform semantic segmentation. So now let's take a bit of a deeper look at our choice of feature extractor. So in this figure, what I'm showing is a random projection of the deep features to the RGB color space. And what this shows is where the RGB values, or where the colors are similar, that shows that those points are also similar in the deep embedding space. So first of all, this is our CNN feature extractor from our contribution 3. And this CNN was trained on coral images. And what we can see here is that it's actually not doing the best job at grouping pixels into segments that correspond with the ground truth mask. In the next column over, we have the Dino V2 model. Here we're using it as a feature extractor. And we can see that it does a better job at producing segments that correspond with what we're looking for. But it does have a lot of grid-like artifacts, and this is due to the vision transformer architecture, which uses positional encodings during training. So instead, we go ahead and use the denoised version of the Dino V2 feature extractor, and we find that this cleans those deep features and improves clustering in the deep embedding space. So now some results for this work, and starting by focusing in on that extremely sparse label setting. So what we can see is our Dino V2 and KNN with the human in the loop approach significantly outperforms. But even if we don't want to use the human in the loop labeling regime, the orange lines show our approach with randomly distributed and grid point labels as well, and these approaches still outperform prior work. What we also notice in this work is that if we compare the use of randomly distributed point labels with grid space point labels, in all cases and across all the approaches that we tried, the grid space point labels outperform point labels. So we do make this as a general contribution in this work as well. Now inspecting the mean intersection union on the y-axis for different numbers of point labels, what we can see is that increasing the point labels from 5 to 100 points results in a significant improvement in accuracy, whereas the benefit of going from 200 to 300 points is far more marginal. So we can also make that as a recommendation. And now we have some qualitative results. So we can see the prior state of the art is the fast MSS approach, and our contribution 3 is the point label aware superpixels. And what we can see here is for only 5 point labels per image, these approaches, their performance significantly degrades. Whereas if we use the denoised Dino V2 model with KNN, we can still produce really meaningful segmentation masks which closely represent the ground truth. So in this work, we've demonstrated that the Dino V2 features clustered in the deep embedding space can efficiently perform point label propagation. We considered that extremely sparse label setting when only 5 to 25 point labels are available. And we also proposed a human-in-the-loop smart labeling regime for selecting really informative point locations. So now I'm going to discuss the collective contributions of this thesis in 3 main areas, starting with the use of these large models, and then I'll discuss the impacts for seagrass and coral monitoring and mapping. So we've investigated 2 different applications of these large models in domain-specific tasks. In contribution 2, we used the vision language model as a pseudo-label generator. And what we found is that using CLIP as a zero-shot classifier did not perform well at discriminating between types of seagrass, but it was really effective in providing a generalized supervisory signal that we could combine with the domain-specific labels to produce a high-performing classifier. And in doing this, we have the added benefit of significantly reducing the inference time as well. And for the Dino V2 model, as I showed earlier, it can be used effectively in combination with point labels if we do that clustering in the deep embedding space. If we try and use Dino directly out of the box on these images, we can see it really fails to perform segmentation effectively, and in the far right example, it doesn't actually produce any segments at all for this input image. So I really think this shows that these models can be effectively leveraged in domain-specific tasks, but in combination with the domain expert knowledge. So now the impacts in terms of the seagrass monitoring and mapping task. So here we established that multi-species seagrass detection and classification as a new task in the literature. And since we did this, we've since seen 15 citations which have used our work and cited our data set. The deep seagrass data set itself has been viewed more than 300 times. So it's definitely improved the attention on multi-species detection and classification in the literature. Finally, we also believe that the approaches presented can enable dynamic adaptation of survey paths towards species of interest. At the higher level, coastal ecosystems in remote areas of Australia and the Pacific have not yet been mapped. But accurately mapping these ecosystems can have huge economic and societal impacts in terms of the blue carbon offsets, improved biodiversity that comes from protecting these ecosystems, and there are also benefits for people's livelihoods which rely on these coastal ecosystems as well. For the coral setting, there are more than 800 types of hard corals alone worldwide, so I think that the application of deep learning models is reliant on fast annotation of efficient model training, retraining, or fine-tuning. So this means going out to a new location, collecting some data, labeling it in a really efficient way, and then quickly getting a model training on those species of interest. The approaches presented here have significantly reduced the point labels needed, and this is by up to 98%. And we've also demonstrated the value of human expert knowledge in improving label efficiency as well. So now looking at the higher level, this year we've had another mass global coral bleaching event declared, and through this event there's really been a clear need for monitoring with high taxonomic resolution identified. So the idea here is if we can monitor with a high resolution, we can potentially see which species are able to recover from an event of this magnitude and move forward with active interventions that play on that knowledge. So we really need to be able to do broad scale, high resolution monitoring in the face of these events. Coral monitoring also has outcomes in terms of improved trust, integrity and transparency in reef management and active interventions. So as we have active interventions and reef management plans in place, we need to be able to prove that these strategies are having the intended impacts, and the way we do that is through accurate monitoring. Now, in terms of avenues for future work, all of the approaches which I've presented here today have considered individual images, and that's predominantly due to a lack of available video data sets. But I'd be really interested to see how we can incorporate temporal information from a continuous stream of images and see how this can improve our performance. I'm also really interested in the task of underwater visual place recognition. So as I mentioned earlier, being able to go out and monitor some active intervention that's been put in place and being able to tell accurately that you're in the same place that you monitored previously or a year ago, that's going to really improve the outcomes for that monitoring. And I think that underwater visual place recognition is a really interesting task that could have more attention. I'm also interested in leveraging the large quantity of expert knowledge that we have available. So we know where different species tend to occur geographically. Potentially they'll have different visual characteristics based on where they are. So if we could incorporate this knowledge as priors in our approaches, that could also improve our performance. And finally, I'm keen to see how these approaches can be applied in other domains as well. So although aerial imagery might not have exactly the same characteristics as underwater imagery, there are definitely still problems that exist where potentially you're trying to detect an invasive species, for example, and you might not already have a whole lot of data that's been labelled for that purpose. So I'd be really interested to see whether the weakly supervised approaches presented in this thesis could be used in other domains. So now to conclude the seminar today, I presented 4 key contributions which have spanned 4 different annotation styles for weakly supervised segmentation. We've considered 2 tasks, seagrass segmentation and coral multi-species segmentation. Our first approach used only patch level labels. We then built on this by using image level labels for that coarse seagrass segmentation task. Our third work looked at leveraging randomly placed sparse point labels in coral imagery. And then we finally considered the extremely sparse label setting in which we only had 5 to 25 points per image. And so we also contributed that human in the loop labeling regime. Thank you so much for your attention today, and I'd now be more than happy to take any questions.
I'm going to kick us off with some questions, and then I'll check in with the panel, and then I'll open it to the floor and to online. So my first question for you, you talked a bit about not being able to do fine-grained segmentation and going into this coarse segmentation.